Okay, so my research is really about gene interaction network and how that's related to cellular aging. So I will first briefly give an introduction on cellular aging, and then talk about my own work. Say I want to use a network model to explain why there is aging, and then provide some experimental results conducted in my own lab and to see why this model making sense. So, okay, so aging is a very important question in biology, but let me ask you, what is aging? What do you think is aging? Give me a philosophical definition, like a textbook.
Aging is like when an organism is in the process of completing a life cycle, I guess, I think of.
Isn't that called development? No. Yes, aging, life cycle seems to imply there's aging and death.
Yeah.
So in a way you say, if something is alive, then it should be dead. So that's, aging is clearly connected to death. If there's no death, there's no aging.
But isn't death included in the life cycle?
Yeah, yeah. But that's not what aging is. Many things have life cycle. This computer is going to go away soon. Okay, what is biological aging? I'm not talking about aging in general. I mean, everything will age. Furniture, papers, right? What is biological aging? What's the difference between the aging of my laptop or this camera with biological aging? There are some key differences. I just want to see whether you can list some which I think are important.
I guess things break down differently.
Okay, how differently?
Like when my last computer stopped working, I guess like it was like a gradual, it got slower, but it like the physical change didn't really happen. Like, but with things that are alive, you can see physical changes and different things that happen as you age.
Why you say when your computer breaks down you don't see physical change?
Because like the structure of my computer didn't change. Like my computer didn't wrinkle or like it didn't shrink. It stayed the same. But it's like the function of the computer changed, not the actual physical computer.
Okay. So there's, well, in biology certainly there are appearance of aging. In computer there are also appearance of aging. If you compare a new computer to the old one, you will see differences. If you just bought a new computer and you have a computer with, I don't know, 10 years old, people probably can, oh, that's a new computer. There's also physical difference between a new computer and old computer. When computers break down, most often they are due to physical damages. Because many of the things are at the nanometer level, you just don't see it on the chip. So, but there's a what if your computer has 2 screens and 2 CPUs? Everything is doubled. Will that help the aging process of the computer, do you think? If you think about if you have an airplane flying, if that's a single engine airplane, that engine fall, unfortunately that plane will just literally drop out there. But if that plane has 2 engines, one of it that it probably can still manage to survive on land. I don't think I'm making a connection.
So, I just don't want to answer any more questions because I keep getting it wrong. So I was going to
No, sorry. Yeah. I'm basically saying there's actually something deeper into that than you think it is. For example, in human, well, in human, you can see the aging process. It certainly appears as time goes by, and in mouse, fruit fly, in plants, grass, but the fundamental those are physiological aging, but the basis of physiological aging is cellular aging. Like in fact, our blood cell goes out in like 80 days on average. What is this? What do you think this is? Rope-shaped single cell.
Bacteria.
Yes, it's bacteria. This is E. coli also age. E. coli, in fact, you grow E. coli in a vial, measure its viability over time. In about 3 or 4 days, they will die out. And what is this thing? Yes, it's a bacteriophage or it's a virus. Do you think a virus also age? According to my definition, actually doesn't age.
But it dies, so...
Yes, but anything dies will age.
But it has aged some since its birth.
Well, let's think about this. You heard about radioactive isotope? Radioactive isotope has lifespan. I mean, like even for C14, its lifespan like I forgot, thousands of years or billions of years. I don't remember. But anyhow, but for some like a P32, lifespan is probably 20 years. If you Google, probably see this. So radioactive isotope also have lifespan, but we do not call that aging. Because if you Google radioactive isotope, see what's the process of radioactive isotope die out?
So what is aging?
Yes, we are trying to work out
I know, but I'm so impatient. I just want to
the question is, there are many things die out. Why, what's the difference? I'm trying to work out a quantitative, precise definition for biological aging, machine aging. I mean, many of you have taken chemistry work.
So it says isotopes decay.
Right,
Like
it's called decay, even though it die out.
Life.
What decay?
The atoms to disintegrate, like the atoms.
Now this is a very simple. Look at the Wikipedia, probably has more sophisticated definition of it. The keyword, this is called exponential decay. It's actually, if you remember from chemistry class, this is called the first order kinetic reaction move. Yeah, basically it goes down like this. It's called exponential decay. So, strength of the isotope, I guess, if you say isotope, and that's time. And what does this curve look like to you? This is called exponential decay with some constant of time. So time is here. So So basically, this is proportional to time, what over time. That's basically what. But what's so funny about this exponential decay is you take it for any exponential function, you take individual from those population and you measure it again, measure at any time again, any time point of exponential function, it's going to look exactly the same as if nothing had happened. So basically those are what we call something, take any time point, those individuals from those with exponential decay, it is as good as new. Because you take an exponential function, you plot it out, it's going to be exactly the same exponential function, decay function. And since all those, if think radioactive isotope, you take the active radioactive type from middle point, it's going to still follow exponential expectation. Any time point you take it, it's always as good as new. That means radioactive isotope is not aging even though they are dying out. Because at any time point, you're going to expect to have the same lifespan on average. Okay? But that's not the same for human. I mean, we take people 25 years old, college, you're not going to expect them to live longer than the infants. That's not the same for human, but that's actually the same for the radioactive isotope. See, what I'm striking here is this is non-aging. The virus is non-aging. And all these are aging. What's the difference is the viruses is always have the same lifespan expectancy even though the time goes by. But on average as a population, they always have the same expected lifespan expectancy. But for complex organisms, including E. coli, the lifespan expectancy goes down when time moves, but the lifespan expectancy goes down for the complex organism. Now the key difference is the chance of dying in this one is constant. So the key is this one. What is chance of dying? The chance of dying is divided by this. Change of viability by unit of time. The T is time. So I have to normalize by the remaining viability. This is normalized. This is, I define this, so for those who are not familiar with the calculus, this D basically means a small slice of change. Right? So when I say if the viability is like this, I say this is basically measure the slope of change. Slope of change of viability. So when I say D viability minus D, it basically measure the rate of change of viability with respect to time. This is basically the slope of change. And then normalized by viability. Viability basically means how much of a, what's the fraction of population is still alive. That's a viability. Now those are the concepts I'm going to put into multiple choice question. And you have to answer that in the back. So viability is basically a fraction of living individual in a population. And D viability over D time, that's measure how the rate of change. So basically this is rate of, So this definition is basically say rate of changing viability and normalized by the remaining viability, the current viability. Viability is going to change from 100%, eventually goes down to 0. I mean, unfortunately, that's just the miracle of life. Everybody will pass away and clear out the room for the next generation and the future generation. That's what Steve Jobs said, actually. So death is probably the greatest invention of life. I think that's really true. So,
so what you just showed us, the slope of change of like the capability of something to grow, like the capability to grow.
I actually haven't mentioned grow yet. We'll probably come back to our topic later. Let's see whether I can make it more clear. So I use the yeast as a model to study aging. So for yeast is a single cell organism. This is also the data you are going to analyze for our project. So yeast is a single cell organism. Basically, yeast is going to divide, one cell become 2 cells, and then 2 cells become 4 cells. How do we measure its lifespan? And for human, you can measure this individual, well, how long it's going to live. You follow that individual, you can measure it. For yeast, a single cell, how do we measure it's going to live? You have to follow that single cell, basically. And the only thing you can follow, and one of the few things you can follow is just measure how many times it divides. You divide once, divide twice, 3 times, 4 times. You just measure how many times it divides. The good thing about the budding yeast, because when you divide it, it's not like E. coli, it divides symmetrically. It just gives rise to a small bud. The budding yeast is like this. When it grows, you're going to have a small bud that then we can count, say, oh, that's 1 cell division. And then it's going to give another small bud. Then we count that's the 2nd cell division. And then it will give another small bud, which is 3 divisions. The trouble is this new cells also going to divide. And so the new cell probably also going to grow bigger and then start to divide. That's going to make us observation very confusing. So we have to surgically remove those cells after this. We call that mother cell. If this mother cell grows, and then we give it a small daughter cell, we count it and then remove it, count it, remove it. So we under microscope, we manually use a needle to remove the daughter cell and count how many times the single mother cell will divide.
So does the number of divisions vary depending on the cell?
That's correct.
Okay. And so that's why we can say that yeast are actually, that's why we can say that they're actually aging, because they have a variation in the amount of, just like humans have a variation of the number of years they can live, like yeast have a variation of the number of buds they're going to live.
You are getting close, except we need a quantitative definition.
Okay.
Just because things going to die out does not mean they age. Like if human and E. coli also follow the exponential curve, that means they won't be aging.
Okay.
Because the funny thing is about so if things follow the exponential curve, say E lambda t, you do a divide of this, so viability is here, you ds dt, you minus s. Since the exponential divided by this is exponential again, so this basically will be ds for exponential. Will be the s for the exponential. And then s divided by this will give a constant.
Okay.
So for viruses, this will, if you, for exponential function, this will be a constant. And this is viability. For viruses, d viability minus dt over viability. This will become a constant.
So the graph would be linear.
It's a horizontal line.
Okay.
It's not just linear. It's
Okay.
it is basically the key of non-aging is the chance of dying. Chance of dying does not change over time. Does not change over time. If things doesn't change over time, the chance of dying doesn't increase over time. It's not, that organism is not aging, because at the time when it's just born, it's going to have the same chance of dying just the next day as it's, say, 100 years old. Right. In human, the chance of a baby to die and chance of teenage to die, it's probably smaller than those people who have lived for 80 or 90 years old. Right. So in human, the chance of dying will increase when we get older.
So with the yeast, as the chance of dying increases, they create more buds? Like,
Excellent, yes. So basically, if yeast divide more, the chance of it, this single cell will pass away, will increase also dramatically. Excellent. See, we are working on the definition now. So for yeast, we can measure how many times it divides. That's actually, technically, we call that replicative lifespan. And we can measure, but yeast also, you don't have to measure yeast how many times it divides. We can also measure yeast, like see how long it maintains viability without dividing. So what's the stage in a cell cycle? What is this? Say it again. I guess so. And this, I thought that's a mitosis, metaphase. M phase is for metaphase. G1 is called I think that's just called M phase. G1 phase, S phase, G2 phase, M phase. M phase has more definition, like I really should remember it. M phase. The interphase M-phase mitosis. I guess that's just called M-phase or mitosis. But if I call that mitosis, it's kind of confusing because the entire cycle is also called mitosis. So I'm still going to call this M-phase. So, but the M-phase has many subtle stages. The chromosome will first condense, then line up, and then separate, goes to the pool. So they are also separate phase for the M phase. But those are called the mitotic cell cycle. M phase, G1S, G2N. So basically you have to remember this. That's probably the most fundamental concept in biology. If after college you want to remember something, that's And G1S, G2N, that's probably those fundamental concepts you should grind into your forehead, probably. So, yeah. But what's G0 phase?
Is that the start of a new cycle?
No.
Oh.
Google.
Oh.
What is G0 phase?
G0 phase, according to Wikipedia, is the resting phase or the period in the cell cycle in which cells exist in a quiescent state?
Yeah, G0 is basically a resting phase. Basically, a cell say, I'm not going to do this cycling anymore, I'll just have a rest. That's a G0 phase, it's quiescence. Or basically say, like if you can imagine a cell like on a cell cycle is like the race car driving on the racetrack. But somehow one car say, I'm going to bail out, have a rest of few cycles, just stay outside of the racetrack, and the cell will just park there and does nothing. That's a G0 phase. It happens very often in single cellular organisms because if you think about it, in natural environment, it's actually not good thing for a cell to keep dividing and dividing, right? Because there's only limited nutrient amount. So the cell pop in natural environment, in the pond, in the soil, in the air, in most cases, cells, those bacteria, yeast, they probably are the G0 phase because dividing is a very costly activity. So we can actually also measure how long cell can maintain viability in G0 phase. That's called chronological lifespan. Basically, we put, yes?
Okay, I'm sorry to interrupt. So when you surgically remove the buds, are they in G0 phase, or is the mother cell in G0 phase? Like
No, when we surgically remove the cell must be dividing cell. That means the cell is doing this cycling.
Okay, so G0 phase is just when there's no cycling at all, like the cell's completely at rest.
Yes, the G0 phase means the cell just stay there. Well, it actually does some metabolic activity, but it's just not going through cell cycle.
So at what point do the daughter's cells begin to replicate?
When the cell goes back to cell cycle. So cell can go to G0 phase. So usually it's in G1 phase it can go to G0 phase, but cell can also goes back to cell cycle.
Oh.
Yeah. So that's an exit. That's a re-entry. Well, if cell can stay at G0 phase permanently, that is a dead cell because it's never going to divide again. Then it's a dead cell. So a permanent G0 phase is basically a dead phase, a dead cell basically. Yeah. So for a live cell, the G1 phase has to be a temporary state. Yeah. So that actually also goes through some interesting philosophy. I mean, some people say pass away, I'm going to have a long sleep. Yeah, I mean, sleep. I found some interesting analogy there too. So, if cells sleep too long, that basically is not a good thing either. So, here's how we measure it for replicant lifespan. And imagine that's a single mother cell. So, and each grow, it have a small bud. And what I do just remove that single cell, single bud, put it aside, and then we just count how many times it has to go through cell cycle. So what do you think the lifespan of this cell? How many cell cycles it has gone through?
3? Doesn't open 3?
1, 2, 3, 4, 5, 6.
Oh. So these little dots are the buds?
Yes. Yeah.
Oh, so what's the big...
That's a single mother cell. That's
So the buds don't actually bud off of the cell, they're still within the cell?
oh, no, this entire, the big cycle, that's a petri dish.
Oh, okay.
Oh, I'm sorry.
Okay, I was sorry.
Okay. This is the plate, this is a petri dish.
Okay.
Then I put a single cell in the middle.
Oh, okay.
Yeah.
Okay, I was thinking.
Oh, okay. Maybe I should make it clear next time. Put an arrow say this is petri dish. Okay. So, yeah. So now this is how we measure chronological lifespan, basically lifespan for non-dividing cells. And what we do is we measure CFUs. What are CFUs? They are not UFOs, they are CFUs. Oh. Yeah, okay. What are CFUs? Let's find out. What are CFU? CFU. Yes, what are colony forming units? So basically, we measure CFUs for a collection of yeast population. We put them usually in water or minimal nutrient. And we measure the CFUs, basically how many cells are alive in that population at day 1. In fact, I call it time 0 because day 1 usually means it has lived for 1 day. So we want to measure the initial time point, that should be day 0. And then after, say, 24 hours, we measure it again. We measure CFUs again, basically. So how many of those cells are still alive after 24 hours? Yes.
So when you do this, how do you keep separate the mother and daughter cells? Are the mother cells marked or?
Okay, this is measure of cell lifespan in G0 phase.
Okay, so you're only paying attention to the
CFUs.
So which cell do you think for that?
It doesn't matter
Doesn't
because well, mother and daughter are relatives.
Okay.
Right.
Okay.
No, this.
So you're not actually trying to see
Right.
how many. You're not actually like measuring a single cell's lifespan. You're just measuring the population growth. From those
Population
cells.
lifespan. Population viability, population growth and population lifespan are different measures. Population growth, it means how fast they divide, how fast they increase. Population viability means how long those individuals can be alive. That's a different,
Okay.
right?
So what are you measuring with this technique?
Measuring
The growth or the life?
viability.
So you're so okay.
We are measuring aging, not measuring growth.
Okay. But what I'm asking, I guess, is how can you measure aging with this technique if you're not paying specific attention to how many cells are coming off of the mother cell?
Because they are in G0 phase. They are not dividing. G0 phase is just Google says it's quiescent phase. Cells are resting there, they don't even divide.
Okay, so are you looking to see how many of them die? Like that's what I'm getting confused about because how is the population going to change if they're in G0 phase?
Because cells going to pass away.
Okay, so you're looking to see the difference, not like the summation. You're looking to see from time 1 to time 2, or time from the first day to the second day, how many cells have died.
That's
You're
right.
not looking to see how many more cells there are.
No, that's growth measure.
Okay, so for this measurement, it's how many cells have died.
Right.
Okay, sorry.
So basically, the number of live cells will decrease over time.
Okay.
If
That
the
makes
number of
sense.
live cells are increasing, then we are doing the wrong experiment.
Okay, that's why I was confused because I understand.
Okay.
Okay.
So eventually we should see, once a cell goes down, actually we should see 0 cell eventually. But we actually don't do that experiment. As long as the cell drop down to below 1%, that's actually good enough for us. Biology working on less than 1%, that is quite accurate. So, okay, this is what we do. This is why we are working this using R. We do it quantitatively. We don't like many other people. They just look at the current, take the average, and say, this one lives shorter, this one lives longer. We do it using this method. We fit into, this is the viability. This is actually a dividing cell. For replicated lifespan, this viability starting population is always 1.0. That means 100%. And that's number of cell division it go through. So viability will eventually drop to 0. Right. So initially, let's say we have 100 cells, all of them alive. And so, what do you think where the average lifespan is? The median lifespan. For the triangle one.
The median or the average? Or are they the same?
They are not the same, but they are very close to each other. What's the median lifespan, let's say? How do we find out? How do we find out the median lifespan? Yes, we found it by, that's very good. We divide the population to half. Why we need half, divide the population to half? Yes, but where is this? Sure. What does the half mean here? Can you read the Alice mark? Well, here's the 50% of the viability. What does that 50% mean? Yeah, so here initially it's 1.0, 100%. Over time it goes down 0% means all like that. What does 50% mean here? Yeah, half the population alive and half the population dead. So that, and the median of 100% is 50%. So the median lifespan of this one is about 25 or 24 something. A median of this circle one, that's about say 35 something. The median of this triangle one is around about 45, 42 something. Now, here's actually, if you think about this, it's actually really remarkable. So, on average, what do you think American live on average?
The average
Lifespan? 75 or something?
I said 77, she said 80.
Okay. I don't know yet, but on average, okay, on average, what's the average lifespan of male, or what's the lifespan of women in this country?
There's a difference. Isn't the lifespan of women longer?
Yeah, can you find out, Google found out? I also would like to know. Average lifespan of a male American versus female American.
Japan has the longest.
Mm-hmm.
Japan has the longest.
Yeah. But there's other story we can talk about later. The number is inflated because many Japanese does not report the dead people because they can continue to receive pension. There's a bias in that too. Yeah.
Okay, for women it's 81.3 and men 76.7.
Oh, let me see. Women's 81 and male is 77. 81 over, so in the US, we have male population 77, female 81. Oh, male, female, sorry. Female, male. So how much difference is this? What's the percentage of change here? You must be excited to see the long lifestyle. Oh, 81 minus 7. 7 divided by 7. 7. 4 divided by 7. That's about 5%. Yeah, I guess about 5%. How much difference is here? Let's assume this one is 44, this one is 35. This one is 25. Let's make it 45. Or something like that. How much difference is here?
How much difference is there?
Yeah. So we have
You're talking about the difference between each of those?
yeah, in yeast. So we have 45. Let's take the average. The first one, 45 or minus 35 divided by 35. I need to dry it before writing. So 45 minus 35 divided by 35. 10 out of 35. About 30%. That's actually a huge difference. And we haven't compared this one. So that's a long-lived, that's a short-lived. A long-lived versus a short-lived. It's like a 20, 25. It's almost like 80%. And in human, you only see this kind of subtle change. So this actually, what this means, it's much easier to study lifespan in yeast because there are such a huge difference. You can easily, the signal is much stronger in yeast. That's besides. You can quickly see how lifespan change. We have millions of yeast cells. You pick a single colony, there will be several million of cells in that single colony. So, okay. So now, what is this? This is something called a Gompertz model. Here's actually the key. After you finish this class, if you don't know what Gompertz model is, I will be terribly, terribly disappointed. Just remember it now. I'm going to ask you to give a formal definition in the exam. Gompertz model of aging. So S is viability. S is viability and T is time. So what is ds over dt means? That means the slope of the chain of a slope of viability over time. That's a slope of viability over time, basically how fast is the change. Does this covered in the calculus class you take?
I'm sorry, I don't remember doing this in calculus.
Oh.
Did anybody else do this in calculus?
What does the slope mean?
Oh, the things like slope, and we did all of those things in calculus, but I don't remember these equations.
Oh, no. I basically want you to understand what is the slope mean.
Oh, right. The
Yeah.
slope is the ratio of rise over run.
Yeah. For example, here, I have a. Let's say I have a viability curve like this. Right. How do this is the viability. This is time. I want to find out the ds dt. How do I find out the ds dt? This.
You have to take the slope of that line at different points.
Aha! Can you come here show us? I forgot how to do it. Can you show me how to do it?
Okay, so this slope, and you guys correct me if I'm wrong, but it's like you take different points of the line. So at this point, what is the slope? At this point, what is the slope? This kind of looks horizontal, so that doesn't look like there's any
Okay,
slope.
that seems to make sense.
And then this is going to level out, so there's going to be a tiny slope here, and then another horizontal line, so there should be no slope right there.
Where are the slope? Well, can you show me where the slope is? Yes, you take this. Where are the slope? Where are the slope?
So I guess you take whatever this point is and whatever this point is, and get that ratio, and then do the same thing for this line.
What do you mean ratio?
Like if there are numbers on this graph, slope is always rise over run. So it'd be the rise of the graph at this point over the run of the graph. It's going to be at this point.
Oh, is that what the
Is that right?
rise and the run? That's what you mean. Run means the run by say run you mean here,
Right.
and rise you mean here. Oh, I see. Okay,
Run
it makes sense. Okay, I see. That's how you always
That's how I was taught to do it. Rise over
rise
run for slope.
over run. That's the slope.
Yeah.
Yeah, it certainly makes sense. Yeah, okay. So, okay, thank you. I didn't know that terminology. So basically you say rise divided by run is the slope. Excellent. So, yes. So basically the rise is here and the slope is there. That's the slope. Okay, thank you. I didn't take my calculator here, so I actually don't know that's called rise, that's called run. So thank you. So, okay. Excellent. So basically, we take I guess I'll draw this even smaller. So basically, calculate all those slopes here. If you right. So basically, here will be. It's actually, first, is this slope positive or negative? Yeah, if we calculate the slope here, a tiny bit rise over run, is this positive or negative? How do we find out this is positive or negative slope? So I guess I can do a comparison. Basically say, is this negative slope? Is it positive slope? So there will be two kinds of slope, right? So we can have a slope which is this way. We can have a slope which is this way. Which one is positive, which one is negative?
Which line is positive, which is negative? The red one is negative.
The red one is negative?
If you put them on a coordinate.
Yes, here. Well, I guess I shouldn't. I should be more consistent. The red is here. Let me put the red is this one. These are red. But what if I have another slope in this one? Basically, we want to figure out, is this slope positive or negative?
Oh, I see what you're saying. The slope should be negative because the line is going down, right?
Yes, right. So the change of this must be negative because things are going down. The change has to be negative. And if it's positive change, it's going to increase over time.
Which part of the graph does the blue line?
There's no purpose. I'm just saying.
Oh, okay.
Yeah,
Okay.
we have the slope is all like this here, but
Right.
the other slope like this, right? The other slope will be like this. And those will be this kind of, this will be a growth. This is aging. So the change of aging must be negative. This slope must be a negative value. But during growth, the slope will mostly positive. And that will be an increase in numbers. Now, here, why do I put, let's see, where do you think this change has the largest number? Where do you think the slope will have the largest value in this graph? If I make it more dramatically, let me make it more dramatic. Sorry. Then you have this graph, and then put the slope again. How do you think the slope will look like in this curve? Maybe I'm going to make this simple for some place. What do you think this slope will change basically? Okay, let's make it a multiple choice. So I'm going to give you several options. Okay, we have choice A, the slope, as a slope. And remember they are all negative, so I'm going to put a 0 here. That's my 0, and everything below that is negative. So I'm going to have this one like this. Those are all 0, 0, 0. This is B, this is C, this is D.
What's the question?
So this is my viability change over time, and I want to see the slope of slope equal ds over dt. Which one is the ds over dt? This is my s. So basically, I'm going to find out the slope here. I want to put the slope into the slope. The slope is also going to change over time. So the s is a function of time, right? So my viability S which is some kind of a function of time. So that calculate ds over dt is also going to give some kind of function of time. Oh, sorry. Yes, those the horizontal one is time. T. And all those are slope, ds over dt. Which one do we think this shape will give? First, should be the slope going down or going up? The slope should be decreasing or is it actually increasing? What? Yes, it does. Because the slope initially is like this, and it's gradually like this, and finally it's almost vertical. So it should be the strength of slope is increasing. Right. So initially it's from a gentle slope, and then it starts to drop very fast. So and then we basically say, well, we should eliminate all those it's decreasing, right? This shouldn't be correct. So then this two should. Right. That's actually a general testing strategy. First, doesn't make sense, you just elimination, right? Choice by elimination. Now then, basically, what's the difference between this one, B and D then? What this one is gradual change, linear, gradual change. This one is not. This one is. This is what is this? It's called exponential. It starts to grow, faster and faster. This is growing, picking up a pace. Why do you think this makes sense?
That is gradual.
Yeah, this is. Initially it's almost flat, so the slope is also going to change very subtly. But then it's going to meet a cliff. It's going to increase faster and faster. So it's really this one makes sense. So we can also put this one. And this is what we call exponential model. Exponential model. In fact, it's not a big deal. I mean, you plot this curve, you can see this. But this is a big deal about 250 years ago, and in fact, someone even presented it at the Royal Society of London. See, here I found out the exponential curve for the population. What did it change? And published a big paper on that. Maybe 20, 50 years ago, it's a big deal because very few people know calculus then, I guess. So, but that's basically what this is. In fact, actually, so the model now, although this actually I ignore a subtle detail here, because let's say if we look at the population change, let's say initially there is 100 person, 100 individual. Initially, if one person died, that's just a change of 1%, right? But what happened at the very end? Let's say at the very end, just 2 individuals left. One person passed away. that's a change of 50%, right? So that's 1 out 2, that becomes 50%. That doesn't seem to look right. Well, this is why we have to divide it by S, because we want to measure relative change. So initially it's 1 out of 100, but we also normalize by 100. And in the end, we also normalize by 50% by the relative size of population too. Actually, I don't think that's actually 0.2%. So 0.02%. Sorry. We measure everything back relative to the initial S, not the in the end. This is also 0.1%, but out of 0.02. That's what it means. Yeah. I think I'm confusing you a little bit. Okay, let me see this. Let's assume there's 100 individuals. Let's start it again. Sorry. I think I started from a wrong angle, that's why. Then I try to correct it, so that's a little bit confusing. Okay, let's look at the chance of one individual pass away, see how the impact on the slope here. So initially we have, let's say, we have one individual pass here, and that's we start with 100 individuals, so that's a change of 0.01 in viability. But at the very end, we also have one individual pass away. That's also a change of 0.01%. If we normalize back here. But it doesn't make sense because initially we have 100 individuals here. But here we have just 2 individuals. So the chance of dying here is just 0.1%, but the chance of dying here is almost 50%. Right. You have 2 individuals, 1 pass, that means it's a half and half chance of dying here. So it doesn't make sense. So both of them means DSDT. That doesn't seem to represent the actual chance of dying, right? Because there are just few individuals here. So if we just measure DSDT, that seems to kind of discount the small sample size at the very end. Right? But how do we correct for that sample size change? Right? Because the entire population is decreasing. If we always count the same number of individuals, even for the same number of individuals change, initially the relative change is slow, but at the very end, that relative change actually becomes much larger. So we have to normalize by the entire sample size over S. That's why we normalize by this. If we normalize on that, so that initially that's 0.99, right? The viability. When one individual passes away, we still have 99%, and at the very end, we have 2 individuals, that's 0.02, and then that's how we normalize the chance of change. Oh.
Is anybody else confused a little bit?
Still confused?
Because if you're not, then I'm not going to ask questions.
Yeah, no, you should. So, okay.
So we're using the slope
for
this equation that's going to give us the, like, what exactly is the equation giving us? Like, what is n equal to?
Okay.
I guess.
So, see whether we can rewrite this. So basically, I'm trying to explain why we need S1 over S here. Right. So we know the ds over dt, that's a slope.
Okay.
And because the slope is a negative value, we put a negative sign here to make the slope a positive value.
Okay.
It's easier to study something positive than negative. So we put a minus here to make the slope positive. But why do we need 1 over s here? Basically, that is the question.
Okay.
So to explain that, let's think about this. Let's say we have this viability here, S, which is the viability. We started at 1.0. It's going to change like this. And then we calculated the slope.
So S, okay. I see what.
That's T. We calculate the slope, so it's going to change like this, calculate all the slope. So it's going to be small, gradually increasing. And then somehow plateauing. Now the question is, if we just calculate this ds over dt and with a minus, for the same amount of change here, and let's say it's going to actually going to drop. Let's now this is going to drop. So let's put up through here. For the same kind of so here we have two values here. One value here, one value here. They are the same value on the ds dt y-axis.
So this is the slope over time.
Right.
Okay.
Plus minus just to make the cost.
Mm-hmm. But where on our graph is the slope positive?
Oh, I take the minus.
Oh, okay.
Sorry. Yeah.
That makes sense.
Right. Yeah.
Okay.
So, but that doesn't. But if you look at, so basically here, let's assume here and here they have the same ds over dt value. But what he doesn't discount is this one, the s is very small, let's say 0.1, here is say 0.9. Even though, let's say, all of them have the ds dt, say 0.05, but for this one, it's only the chance of dying is just 0.05 out of 0.9 here. Right. But this one, the chance of dying is 0.05 out of 0.1 here. So this is almost 50%.
Is 0.05 coming from?
0.1, yes.
Okay. So what you did was, so is this your 1 over s? These values, 0.05 over 0.01?
Yes.
So
that's why we put 1 over s. This is basically my s value here.
Okay.
This is my s, this is my s. And that's my ds dt minus. Basically,
okay, so what you're doing is you're correlating both graphs. So you have 0.05 from the graph that shows the slope, and then you've got the other value from the actual viability graph, right?
Excellent.
Okay.
Yes, right. Basically, that's how do I calculate my chance of dying? This is my chance of dying. As a function of time. But how do I calculate that one? It's basically minus ds dt divided by s. Maybe that.
So you have to have both graphs to be able to figure out 1 over s.
Visually, yes.
Okay.
So you calculate the ds over dt, and then you just divided these two graphs.
Okay.
Okay, anyone else have question? Please ask. Okay, let me explain. Okay, so it's good we really get into this. So this is my model of chance of dying, and I'm going to say the increase of chance of dying, give the formula that's called mortality rate. This is also called mortality rate. And so what happened 250 years ago is just someone say, oh, what I found is mortality rate changing is really an exponential function. And once that person published the paper, people going to call that the last name of that person is called Gompertz. And then people just say, oh, this is a Gompertz model. And we even just to give credit to that person, we also call the coefficient called G for Gompertz. So that's T, that's G. And this is just the initial rate for as a constant. I probably should change that to M0 as more consistent. But I call it R0 because R stands for rate. But it probably also makes more sense if I just call it M0. If I call it M at the time 0, probably also makes more sense. And the funny thing is, if we just have this definition, then we can kind of back calculate what kind of viability can we get. And this is basically solve this. If we have this alternative model definition, and the viability is basically this, you can actually solve that one. Solve this differential equation, and you can get this definition of viability. How do you solve this one? You basically do an integral on both sides. You can solve it. And what does this mean? This ds divided by 1 over d, this is basically d log ns. Right? So basically that means d log ns over time equals R0 exponential dot t. And then you can just put the dt on this side and integral on both sides. So that I'm going to have d log s equal minus r ddt dt. And then just do an anti-integral will be log s equals to that. This whole thing. And then you know this is going to be go to exponential part, and that's this part. And this part will come to the part. And d log ns will give you the exponential part. So I'm going to ignore that. If you want to find out, you can just review your calculus and calculate it. So now you know. So you don't have to know that. All you need to know is if we have this Gompertz definition, the viability actually can also be known. That's how we actually can fit if we draw a curve, how do we know the Gompertz parameter? We just fit that curve with a viability curve. And then we can know all those parameters there. So okay. So here's my formal definition of aging basically. Increase of mortality rate over time. If that is increasing by time, that's biological. That means aging. If it follow exponential model, that's biological aging. This is my definition of biological aging. Gumper's model for biological aging. But in general, if the mortality increase as a function of time, if this is the increasing one, I will call that aging. If the mortality rate is a constant, that will be a non-aging. Basically. That's actually now I have a formal definition to describe what is the aging system, what is a non-aging system, and what is biological aging, what is non-biological aging. So the biological aging, I say, is exponential increase of the increasing of mortality rate over time. If it's not this exponential form, I call that non-biological aging. Non-bio aging. If it's other forms of aging, right? So then, basically, I have a measurable way to say, this system is aging, this system is non-aging, this system is biological aging, this system is not biological aging. So what I have, basically, I have a system to say, this is I will have a theory or model to say what is computer aging, what is the, say, the change of human population, or change of, say, the dog population, the wolf population. So in fact, this is something you should remember. This, I hope you at least learn a few things here. What is DS? What is the derivative slope? What is the positive slope? The negative slope? And how we can normalize that slope? Those are actually also a general principles, not just for aging, for many things. So, if we apply that concept to the viruses, here's what we see. That's the for viruses, how do we measure its viability? We measure it by the infectious ability. Sorry, what?
Is that kind of its fitness in a way? Like,
yeah, that's basically viability for viruses. How much it can infect the host. If a virus cannot infect host, it cannot live on its own. That's a dead virus.
So does it matter?
Yeah, it's something you think how do you tell an organism is viable or not? But virus doesn't live on its own. How do we measure viability? You measure how it can make itself livable. Infectious rate is how. Yeah. So, but basically, viruses have an exponential viability curve. You do a DSBT normalized S over time. That means viruses have a constant mortality rate. And so this is basically a plot of a log. This is basically a plot of log viability, and that's time. So we basically plot a log of S over log of E minus R0T. What is this? A log of E, that's basically this part disappear. That means minus R0T. And how does minus R0T look like? Just like it goes down, right? Basically here. Down 0 goes down like that. And that's exactly how the viability of viruses look like. So viruses, based on actually not my definition, this is actually a common definition. That's a French group working on this. So then we know viruses is a non-aging system. Surprising, isn't it? we actually have a species that doesn't age. We have a species that doesn't age. In fact, viruses is probably the most ubiquitous species on this planet. So in fact, the most abundant species on this planet is a non-aging species. That's quite surprising, isn't it? Yeah. It's very provocative when you think about it. Now, what kind of question will you are you thinking about now? Based on this.
So
I wonder what you are thinking right now. So we have a non-aging system. What do you think we can study from here?
how to fight like anti-aging.
Okay, anti-aging, yes, right, yes. Yeah.
I guess too, if you have something that doesn't age, like, are you asking what can we use this virus in order to study? Or are you just
Precisely.
okay, so
Yes.
I guess if you have something that ages depending on what your, or something that does not age, depending on what your experiment is, I guess that would be beneficial because you have more time to actually watch what's happening in the experiment.
Yes, I'm actually, well, I'm actually curious, what do you think, what you can do? If I tell you something is non-aging, what kind of things do you want to know? What kind of question you have in your mind? What do you want to study next? What kind of problem you want to address? Okay. You want to see what's the genetic mechanism between aging and non-aging? What do you want?
That sounds right.
What?
I said, I think that sounds right.
That's right. I also think that sounds right. In fact, my title is called genetic model. So excellent. So you think my project? Can't you think to make some sense? No. It certainly makes sense. Here, from gene network. Yes. Yes. So, well, to me, it's like we have a viruses which is non-aging. We have cells which are aging. Basically, the natural question: how can we with all these genes? I mean, genes are genes. How can we have species that age and that those don't age? Why some species tend to have long lifespan, some species have short lifespan, and how for most people, we probably also want to know, can we live longer? Yeah. So basically, those kind of questions naturally come to most people's mind.
To find out how to make things last instead of trying to live longer. Because I mean, if you get rid of like morbidity effects on that eventually, you live longer.
Yeah, I think that's a natural. Yeah, that's a good target. That's a good if you can get rid of. Yeah. In fact, that's what most of the methods work. How to prevent aging. So, well, for yeast is a model organism. If you remember from 125, you use the yeast as a model to study. DNA mismatch repair. And when DNA mismatch repair have defect, genes will tend to have more mutation. When mutation hit cell cycle control, and cells can go on dividing non-stop without control, and that's what cancer occurs. Uncontrolled growth most often it leads to cancer. That's unchecked cell cycle. But this is part of but I studied this used to study aging, but we actually sequence the yeast genome. In fact, we have now called every gene in yeast. We know many of them will influence lifespan. But my question is, which gene is the cause of aging? In fact, there is no such gene exists. There's no cause, there's no molecular, there's no gene we can say is the cause of aging.
It's a network.
Network effect. In a way, yes. Well, I kind of despite, I guess since I started with gene network, I cannot tell you what I'm going to. But despite the tremendous amount of influence on aging, there are some kind of called universality on aging. Yeah. Universality. What kind of universal principle have you learned? Well, that's absolutely true. Some general principle. Aging itself is just a phenomenon. What's a general principle? Universal principle you have? Can you tell me some?
All living things are never slaves.
Okay, that is not 100%. Viruses that is not made of.
They're not living.
Virus is not living. If you have a virus infection, your virus is not living. If you have a flu, you sneeze.
I was always told viruses aren't living.
Okay, who told you virus is not living?
Maybe it was easy.
If virus is not living, we shouldn't even have a flu shot any year. Okay.
The virus is not living. Because they can't produce on their own. So that's what is it? Was that the cell? Is that the cell?
But many parasites, they cannot live on their own. Right. Right. Yeah. In fact, mitochondria used to be bacteria and doesn't live on its own, but right. Yeah, we humans also cannot live on our own if you get rid of those nutrients. If we don't eat plants.
If we don't what?
If we don't eat plants, if we don't depend on other species, we cannot live on our own either.
That's not a tenet for living, though.
What?
Being able to survive on your own is an tenet for living. Like, isn't it like reproducing, independent reproduction?
Okay. I'm going to surrender. Let's say that is the general principle. Most of the cellular organisms are going to have a cell to live with. Non-cellular organisms actually no.
Okay. Well, let's see another principle.
Yeah. Similar to.
Is that be biological?
Yeah, biological, anything, many principles. Yeah.
The central dogma, I guess.
Yeah, central dogma is also, yes, that is indeed. Central dogma, we have DNA to mRNA to protein.
Wait, how is that considered a central dogma? What if it doesn't have DNA? I mean, how's that compare to universal? But everything doesn't have DNA though.
What things doesn't have DNA?
RNA viruses.
How does the RNA viruses work?
How do they work?
Yeah.
RNA.
Because it goes back, use a reverse transcription, goes back to DNA and then use DNA.
Yeah, but doesn't it only have RNA?
I think it's not really viruses.
What about the Newton's law?
What about Newton's law?
That's a universal one. Right. Anything, even things inside of the cell still follow Newton's law. It's just so tiny that it's probably negligible. But certainly individual follow Newton's law. You want to walk out using stairs, not just jump out. So if Newton's law doesn't work in biology, we can just fly out our window directly to the pen. That's there is actually another phenomenon we have been talking about a lot in this class. It's something called randomness. We're always talking about random noise. We're always talking about bell-shaped distribution. That is called normal shaped noise. Normal distributed noise, and that is a universal.
That's universal.
Yeah, like a height, you measure people's height, seem to follow normal.
Okay, so that's universal.
Yeah.
How is that universal?
Yes, why is that universal? Because there's same kind of randomness, random movement in many processes.
So like, are you saying everything can be put into a normal distribution? Is that
No,
what you're saying?
many things follow normal distribution. It is one of the ubiquitous phenomena. Yeah, like if you draw the lottery numbers, not simple follow. Right? If you look at measure the height of trees, you measure the weight of people. In fact, if you
something normalized though, as far as the work.
no, you don't. Well, sometimes if it's not normal distribution, you can normalize it back. You can transform it into normal.
I feel like if I measure a bunch of trees out here, just to select a random population, they're not all going to have the same like E mode and EF for it to be normal. Because isn't that like under requirements for it to be normal, that all of those have to be selected?
For large drawing, you will be. Actually, how about you try if you want?
Okay.
No, you can just randomly draw. Look at some. Yeah, there are so many things on normal distribution you will be. In fact, in the old days, there's a biologist, I forgot his name. He found out that there's a bell distribution of the seeds, maybe wheat, and then he measured the height of some kind of graph. It's also bell-shaped, and his conclusion, oh, I must see God, because everything follow the same shape. This must not work out. This everything is well-shaped. Yeah. I forgot his name, but yeah. But not everything is normal. Like the lifespan, it's actually, well, we basically say lifespan is Gaussian distribution. The Gaussian distribution is the normal distribution is a symmetric. It's bell-shaped symmetric. The lifespan is actually asymmetric. It's actually, if you plot it out, it can be approximated by normal distribution. That's why normal distribution is this bell-shaped curve can also used to approximate not exactly normal distribution like lifespan, even though I believe it's a jumper, but most biologists just don't even care. They just say, well, we plotted the bell-shaped curve and use that. So it's good enough for at least some of the work people have been doing. But let's say why there are these normal distributions everywhere. Even though tree height, student test score, and lottery number, they are not even the same thing. Why they follow the normal distribution? I mean, they're not even the same thing. Why they can follow the same shape of bell shape distribution? Basically, you take distribution. Basically, you take all those individuals, do a histogram. It's going to look like bell shape, right? I mean, we in our class we kind of basically I think we did a reading ability, shoe size, so that's what it's normal distribution. It basically assumes they are, even though they are completely different entities, but they follow the same kind of random movement, randomness. That's actually the fundamental reason. Basically, for things to follow the normal distribution, there's kind of a random process. And this kind of random process occurs in many different fields, for many different subjects, even though they are completely different things. But this randomness are the same. That's basically it. So, but my question is, my point is, in aging, there's also a universal observation. In fact, in human, we see there's a log correlation between a Gumpert parameter. I'm going to tell you this, that we probably have to come back next time. So this is, remember in the Gumpert model, there are two parameters: initial mortality rate and Gumpert's coefficient. Gumpert's coefficient basically measures the increased mortality rate over time, G. We can use that as a proxy of rate of aging, G. So, so this will be the log of R0. Sorry. If we plot this one out, the log of R0 over G, in human population we see a negative correlation line there. And in yeast we also see a negative correlation line there. In fact, this negative correlation also in many species, in fruit fly, in mouse, human, I'm sure in worms, it occurs in almost every species we have observed, which have about the aging rate. I'm going to ignore this. Actually, I try to explain everything here. I try to also come out with a general principle to explain why there's aging. Since my main, I think Jessica already know what I'm trying to get. I basically think aging is caused by gene interaction natural. But in general, if I want to generalize, I think aging is a property of biological complexity. I think I'm going to end here.